Hi all, welcome to the NOAA Omics seminar series. We'll get started in about 5 minutes.
Hey, Nicole, I don't see the share my screen anymore. Is that something I'll get back when you give it back to me?
Yep, you'll get it back after I finish up my 2 introductory slides.
Awesome. And then I also don't see the questions block right now. Will that come up later?
I believe so, and if it doesn't, I will read them aloud to you and can repeat if I need to.
Awesome, thank you.
Thanks, Steve. Alrighty, hello everyone. Thank you for joining today and welcome to the NOAA Omics seminar series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the past few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway, both within and outside NOAA. The seminar series The seminars from this series are made available on YouTube and posted on our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Publishing Omics Data to GBIF-US and OBIS USA by Dr. Steve Formel. I'll go ahead and share presenting. Alrighty. Stephen is a biologist at the USGS and the US Node Manager for OBIS and GBIF. And Stephen, can you, do you have abilities to share? Great, okay. In this role, he serves as a liaison and educator for data mobilization, best practices, and the standards used by the OBIS and GBIF communities. He is a microbial ecologist by training, with past research focusing on the relationship between salt marsh grass, its microbiome, and the deep water horizon oil spill. Prior to working at USGS, he was a data officer at NCEI and worked as a data management lead for the MDBC project, and also served on the NOAA Omics Data Analytics and Bioinformatics Working Group. Thank you, Steve, for presenting today, and I will now pass it off to you.
Okay, thank you, Nicole. So you can see my title slide, okay?
Yes, we can see your slide and hear you clearly.
Okay, perfect. So, yeah, today basically I'm going to go over some of the basics of what GBIF and OBIS are and how the US nodes, relate to all of that, and then also go over some of the basics for publishing eDNA through these initiatives, and share some resources to help you get more into that. And I'm actually going to start with that last bit, the sort of the dessert of the talk. So this is the kind of slide that I think a lot of people have been looking for a while, so I finally took the time to make it, where here in the first couple columns, we have a variety of links going to background on OBIS and GBIF US, as well as GBIF North America. And then the middle two columns are also training and guidance, both courses you can take and also things like workbooks you can use and other types of reference material. And finally, on the right-hand side, are various community support resources. So everything from Slack groups to discourse, and also just general emails that you can reach out to. I want to emphasize that, my job is to help the US scientific community. So, no question too small or too large to ping me with. And then another thing that I think a lot of people find useful for learning how to use standards and publishing platforms is to have examples. And so here today, I'll primarily be talking about Katherine Silliman's data set that she published this past year to OBIS and GBIF, the one at the top, the GOMEC data set. But there are a few other good examples, and the one on the bottom is actually an example of publishing qPCR data. So, so you can use this as a place to explore how others have published the data and get a sense of how you might be able to do the same with your own data. So the place to start, of course, is talking about what exactly are GBIF and OBIS. These two acronyms can be a bit of a black box if you're not part of the biodiversity science community already. GBIF and OBIS are two international initiatives that are aiming to provide open access to biodiversity occurrence data. And so a lot of people first encounter them through their role as publishing platforms and data aggregators. But in fact, I really want to emphasize that these initiatives represent a few decades of investment from the global community, and also a vibrant community of the nodes, the publishers, and the users of standards and practices. So it's a place that you can get involved with, not just publish data too. And so GBIF stands for the Global Biodiversity Information Facility, and its data holdings are a little over 2.6 billion occurrences of biodiversity. And so this corresponds to over 100,000 data sets and over 2,000 publishers, and can be linked to at least 10,000 peer-reviewed papers. OBIS, on the other hand, is the Ocean Biodiversity Information System, and because it's a marine-only database, has smaller data holdings, but it's still quite impressive at over 120 million occurrence records, accounting for over 4,000 data sets and almost 150,000 taxa. Like I said, these are really, these are networks, these international initiatives. And so although they are both headed up by secretariats, they are really strong communities that are dispersed throughout the world and are quite diverse. So here on this international map, we're looking at, in blue are the countries where there's at least one GBIF node, and then the yellow points represent OBIS nodes. Now the places in gray, that doesn't mean that nobody participates from those places, it just means they have less formal involvement than the places in blue and yellow. And this talk is about GBIF US and OBIS USA specifically. And so these are the US nodes for both GBIF and OBIS. And I'm the node manager for both of these nodes. And I work in the biogeographic science branch of the US Geological Survey's Science Analytics and Synthesis. And we've been managing these nodes for some time. Recently, we also brought NOAA's IUS in as staff for OBIS USA also, and that's been huge. We're working with Matt Biddle from IUS. And then I also want to make sure I mention that GBIF US is funded by the National Science Foundation and also includes collaboration with many other scientific organizations in the United States to help guide that effort. Generally speaking, what we're doing as the US nodes is trying to represent US interests in GBIF and OBIS. So we're also here to advise the scientific community doing things like I'm doing right now, which is trying to help people learn about biodiversity informatics, data, and standards for sharing that data. We also work hard to help mobilize and synthesize data to both enhance its fairness and to increase its relevance on national and international scales. And the last point I want to mention at the bottom there is that a lot of people first come into contact with us when they want to publish through our integrated publishing toolkits or IPTs. These are essentially web applications for sharing your data, and we run two. We run one for OBIS USA and another one for GBIF US. And so why do we, the US nodes, want to work with GBIF and OBIS? Well, back in 2022, Terry McConnell very quotably said, sparkly fountains need robust plumbing systems. And what he meant by this is that, if we want there to be shiny, sexy science, it needs to be supported by strong data management. And the US has been a voting participant in GBIF since 2001 and has been with OBIS USA since 2005. So the USGS, and now also with NOAA IUS, is shepherding over 2 decades of investment in that robust plumbing that we need for useful national and global biodiversity science. And one of the things that I think people frequently sort of bump into and they're not sure what to make of it is there's a lot of similarity between these organizations. And so a big question is, well, how are they the same and how are they different? And how are they different? And one of the points that I want to emphasize is that they're definitely not at odds with each other. These two groups, like many international science projects, work hard to collaborate and have their work be useful together. And so both of them, both OBIS and GBIF, are active and energetic communities that have been working to develop, maintain, and promote standards and practices for over 20 years. They both promote open data and science and publish massive amounts of data through their platforms. Importantly, we are now in the middle of the second letter of agreement between these organizations that recognizes that their respective communities will benefit if they work together. And we are now developing a stronger action plan, which should be coming out later this year, which gets even more, which works to align the two groups even more. And the last part, which is maybe the most important to the normal user, is that GBIF and OBIS are built on the same data and metadata standards. So this is Darwin Core and ecological metadata language primarily, and they share these open source tools. And so that provides a lot of benefit to users because practically speaking, their data are relatively interoperable and reusable between the two. Some important differences to know is that GBIF has a global scope, but OBIS only includes marine occurrences. And if you know about the funding, I think that will help you appreciate some of the governance behind these organizations. So GBIF is funded primarily by individual participant countries. For example, we fund GBIF US through the National Science Foundation, and OBIS is funded by UNESCO, an agency of the United Nations. I also want to point out that we're a bit unusual in this country because GBIF US and OBIS USA are managed by the same people. In other countries, they might be managed by different people or different agencies, or they might only have one node or the other. And so this is important because it works really well for the United States, I think, to have both both initiatives in one place and increase the connectivity of our science community. However, I just wanted to point out that that's not the case in every country. And you shouldn't be confused in thinking that GBIF and OBIS are run that way everywhere. Okay, so let me tell you a little bit about how the data flows, because I think this will help you appreciate the different parts of the process. So this is a diagram from GBIF, but OBIS is fairly similar. So starting on the left-hand side here, we have data coming from all types of entities, so museums, universities, citizen science, and they're creating that data in all kinds of ways. So they might be collecting specimens, they might be observing it or using literature, or it might be remote sensing or DNA, like we're talking about today. And those data, importantly, are shared through open standards like Darwin Core and AOML, and then licensed through open licenses through the Creative Commons. Those data are then published through the IPT, the Integrated Publishing Toolkit that I mentioned before. And in this way, they are shared with the global databases. Those data are then served through things like through cloud services and through web browsers and also through APIs. In the case of GBIF, they also serve their API up through hosted portals. So we have gbif.us and GBIF North America as community-specific portals, and other thematic portals can be created. And then finally, I want to impress upon you the diversity of use cases for which these data might be used. I think a lot of times we as scientists have sort of myopia when it comes to thinking about how our data would be used by others. We tend to primarily think about it for its primary intended use, or why we collected the data in the first place. But I think that after people publish to OBIS and GBIF, they're often surprised to see their data being used in other countries and other places for purposes beyond its initial intent. So for things like climate science or human health or food security, things that might not necessarily be what you expected. And I think that's one of the biggest things to highlight about publishing to OBIS and GBIF is that by doing this, you're really opening up your data to a global community for that community to use it in ways that you can't imagine. And so to put a point on all this, so what do GBIF and OBIS do well? Generally speaking, they coordinate and serve interoperable observations of biological occurrence. They promote and catalyze progress for open science and fair data. And importantly, their platforms allow you to search data at the occurrence level, unlike many repositories, which only allow you to search the data set level or the metadata of the data set. I'm not going to get into this today, but both groups do also track data use, and GBIF does an excellent job of also tracking citations. That will be even more improved actually with OBIS as well, coming soon. But this is I think an important thing to convey, because both GBIF and OBIS are well aware that citations and getting credit is the currency of science, and so they work hard to make sure that people are getting credit for the work they do and the data that they share. Both groups also publish many tools and guidance to help you self-publish. And again, I want to come back to the fact that this is a very active and very diverse community. You can get involved with any of these pieces and give input for improving it, or helping overcome challenges that you encounter. And I think by doing so, you really might open up yourself to understanding more about open science, and also, again, the diversity of ways in which people use different people use the same data. A couple things in development that I want to mention that are specific to eDNA communities and the omics community is, we're well aware of the challenges of taxonomic dissonance between taxonomic databases. And so people are working on ways to help align these taxonomic databases so that no matter what reference database you use, the taxonomy is making sense across data. Similarly, we're trying to develop ways to facilitate round tripping of the data for constant improvement, especially with regards to taxonomic updates. We're interested in adding sequence searching as a way of identifying data, not just searching for taxonomy. And then also GBIF is developing a simple tool for non-technical and sort of small to medium-sized DNA publishers. I've linked to it here. This is a graphical interface where if you're not necessarily very tech savvy, or or you might just have sort of a one-off data set. You can transform your data and reshape it into that the right structure to publish it as a Darwin Core archive through GBIF. And then finally, I also want to let you know that we're working hard to expand our US node staff. We've been getting a lot of requests in the last couple of years to help support various projects, and it's evident that people are excited about what we do and they want to use it, but that's of course going to require support. And so I just want to make sure I say that we're working hard to provide that support. And so at the end of the day, why use GBIF and OBIS? You know, it's hard enough publishing data as it is. I don't think you should strive to make it any harder for yourself than it needs to be. These are off-the-shelf platforms for publishing and tracking biological occurrence data with really minimal financial and staffing investment from you or your lab or your project. And I want to point out that USGS and Springer Nature accept GBIF as an archival repository, and also we've set it up so publishing to OBIS USA satisfies NOAA archiving requirements by having those Darwin Core archives also harvested by NCEI. And so the take home here is, you know, this mountain that we're climbing to publish our data and make it fair, if you can, start here in the middle. Don't start here at the bottom if you don't have to. Okay, so another question that comes up frequently is, well, if I invest in these things, how do I know they're not just going to go away, you know, next year? And that's a fair question. That happens all the time in science. And I would say that one is you can draw confidence from the fact that these two initiatives have been going for a few decades, but also, you know, and this means that both organizations do rely on regular international investment for funding. But OBIS, for example, recently transitioned from an IODE project to a program, which means it's now receiving core funding and staff support, making it even more stable. And also the open standards in science that they're built on means that even if there was a catastrophic collapse, migration of data and standing it up again somewhere else by somebody else would be relatively simple if there was that catastrophe. Similarly, in the US, you know, I've told you we're trying to expand our staff, but we are relatively small. And so it's logical to ask, well, what happens if that goes away, that support? So GBIF US has been funded by the NSF for some time. And there's a US delegation that is well aware of that funding and also the importance of that funding. For operation of both nodes, the USGS is funding the node staff salary and travel, and then NOAA IUS is now also contributing to staff to OBIS USA. But the IPTs are actually hosted by GBIF, and they could easily transition to other management and continue operating without any additional US funding. And also, I want to say that both secretariats have funding and training in place for new nodes and new node managers, if it was the case that again there was some sort of catastrophe or other reason that that what's working right now is shut down. Okay, so now we're going to actually switch over to from all this background and context to what it's like to publish data, and specifically eDNA data. Now, generally speaking, it can be broken into 4 parts. You collect the data. If you're already using a standard like Darwin Core, great. But if you're not, you need to align to Darwin Core, and then create a Darwin Core archive. Typically, you do this through the Integrated Publishing Toolkit, or IPT. And then you need to register that Darwin Core archive with OBIS and GBIF so they know to harvest it. And again, you typically do this through the IPT, although there are other ways to do it. With DNA-derived data like eDNA, there are of course a lot of extra metadata, sort of molecular biology metadata that you're interested in capturing. And so it's worth pointing this out at this step that this is an understood concern, and there's also mechanisms for dealing with it. So at the top there, letter A, this is a typical observation. So let's imagine that you collect a fish, you visually identify it, the taxonomy, and then you publish it through as a species list, for example. With DNA, you might collect some water, and then everything in the dash box is all of those molecular analyses, the wet lab and the dry lab work. So extracting the DNA, sequencing it, the bioinformatic analyses, resolving it against reference libraries, and all of that happens, but ultimately what comes out the other end is no different than what happened in letter A. It's just a collection of species that occurred in a certain time and place. And so the way that we're going to capture all this information is through 2 standards. the Darwin Core standard, which helps you describe those biological observations, for example, ASVs, and then also ecological metadata language, or EML, which is helping you describe the entire data set, so the entire collection of all your samples. The way this data is shared, the unit of Darwin Core is the Darwin Core archive. And so you could think of this as sort of like a lo-fi netCDF, if that's something you're familiar with, but essentially it's a zip archive. And this is what is harvested by NCEI when it archives the OBIS USA IPT. So in green at the center there, we have a flat table. By this, I essentially mean a spreadsheet where the column names are going to be Darwin Core terms. And then in yellow are various types of extensions. And so those extensions describe important attributes that might not be common to every data type, but are specific to certain data types. So for example, the DNA derived extension is one extension that you might include to describe the extraction and sequencing and other molecular processes. And then the extended measurement or fact extension would be where you could describe things like environmental chemistry or experimental covariates. In red, we have two metadata files. The one on the left is a metadata file that's small and most people never need to worry about. It's called meta.xml and describes the contents of the Darwin Core archive. And the one on the right is the ecological metadata language, or EML metadata, which describes the entire data set and is structured as XML. It's worth saying that EML and XML, although they both end ML, it has nothing to do with each other. And that's something that beginners frequently get confused about. XML is the structure and EML is the standard. Again, all of this is then zipped up. And this is something that I could send to you through email. You could open up, and because it's based on an open standard, you could immediately use without having to question anything about it. Getting into the DNA derived data extension, this is the extension that expands occurrence data with all that molecular biology metadata I keep mentioning. And so basic Darwin Core involves terms to describe events, locations, this sort of standard information across occurrences. This extension includes terms that come from other standards, so the GGBN data standard, the Genome Standard Consortium's MIXA standard, and also the MIKE standard. And essentially what it does is put them into one place in a syntax that works with Darwin Core. Now, guidance on this is available through this Publishing DNA-Derived Data through Biodiversity Data Platforms guide published by GBIF that has also a section specific to OBIS in it. And I want to say that if after this talk, if you were going to start somewhere and you haven't encountered this document before, I would encourage you to go and browse it. It's probably one of the things I come back to the most when I'm working with DNA data. It has a lot of contextual information and also specific examples of how you may want to use certain terms. Okay, so in the abstract for this presentation today, I said I was going to use examples from Silliman et al., which is Katherine Silliman from AOML. She published the data from the GOMEC 4 cruises the data was collected in 2021, and it was just published to OBIS and GBIF this last year. On the left-hand side is the landing page at GBIF, and so there's a QR code for it if you want to go check that out. And on the right-hand side is the landing page at OBIS, also with a QR code. And so I wanted to give you these landing pages so you have a sense of how that Darwin Core and how that EML gets served up through a web browser. In my opinion, they're both excellent user experiences, but also importantly, if there's something you don't like about it, please give us feedback. We'd love to improve it and make that user experience better. The data is essentially equal between the two places. Data served through OBIS and data served through GBIF aren't effectively different. However, these two spaces are catered to their specific communities, and so some people might prefer one or the other. Also still using Katherine's data set, I want to show you what that Darwin Core archive looks like in real life. So up here at the top, we have a screenshot of the archive unzipped. So this is a full eDNA study that includes over 311,000 ASVs. Those are the occurrences, which are the ASVs broken out over samples. This is a map of where those samples were taken. So again, I just want you to appreciate that this is a substantive study and not a toy data set of any kind. Up here at the top, this is that expanded, the structure of the Darwin Core Archive that I described before. So we have our occurrence.txt. This is our table that has Darwin Core terms, the occurrence core terms as the headers. The DNA derived data.txt. This is where our DNA derived data extension terms are stored. The EML.xml is where the ecological metadata language is described, and then the meta.xml is where some additional attributes about the Darwin Core Archive itself is described. Okay, and so now what I'm going to do, just to sort of wrap things up, is walk you through some specific examples so you can get a sense of what those terms look like and how they were used in the Silliman et al. Data set. So most importantly, there is this term occurrence ID, and this is the identifier for that ASV in that sample. And you notice that it's long, it's unwieldy, but the important point is that it's unique and it's persistent. So this is the key that we use to describe all these data. Everything else can be updated. So let's say there were revisions made to the taxonomy or the location or something like this after it was published. That's not a problem as long as that occurrence ID remains stable. Zooming in a touch, we see some other terms that basically describe the where and the when of these data. So we have the term event date, the Darwin Core term event date, which is the timestamp of the observation. We have a couple other terms describing the location. So locality is a literal term that can be very flexible to your needs. But then location ID can also be something like a station ID and be an identifier that you're using across other studies. The coordinates are described as decimal degrees, with the geodetic datum being WGS 84. And then finally, there are two terms to describe depth: minimum depth in meters and maximum depth in meters. In this case, they happen to be the same, but you can describe a range if that's useful. Before I go any further, I want to point out that there's over 200 Darwin Core terms and then another few dozen in the DNA-derived data extension. And so the point is not for you to learn all these terms, but rather to learn to use the reference material that helps you be aware of them and figure out which ones you should be using. And that reference material is in the resource slide that I showed at the beginning of this talk. Going on, we can get into some of the more DNA-related terms that are used. So again, we have that occurrence ID, which is linking all the data together. And here we're looking at organism quantity, which is the number of reads for that specific ASV in that sample. Here, the organism quantity type is described as DNA sequence reads, which is a community agreed upon piece of vocabulary. But I want to point out that this is very flexible. Organism quantity and organism quantity type can be really changed to whatever makes sense for your needs. So if you have something that's not necessarily, for example, eDNA data, you can use other units. This is not a matter of it only being presence absence or individual count or something like that. Then next up, we have sample size value and sample size unit. And so this is the total number of reads in that specific library. And then finally, zooming back out here, here we have associated sequences. So these are the repositories where the raw sequences are stored at NCBI. And again, there is some flexibility here in how to describe it, but the point is to try to make it easy for users to find those raw sequences should they want to go back to it. In this case here, we have identification remarks. This is the ubiquitous sort of comments field that most biologists have in most of their data. And one of the things I want to point out here is Katherine decided to add a note discussing the limitations of the taxonomic assignment strategy in case that was a concern for anybody working with this data. Finally, going down here, we have verbatim identification, which is the original identification. So this does not have to be controlled. This can be however it comes out of your reference database or your algorithm or what your person in the field described it as. But then we have scientific name, which is a controlled term that in this case aligns to the worms taxonomic database. And we know that because the scientific name ID uses the worms LSID identifier here. And so this way it's unequivocal which Karenia brevis is meant when we are describing this ASV as such. So I want to show one similar example where everything here is looking very similar except for this information at the end, because this is an important one for the omics community. You'll notice here that verbatim identification, so what came out of the taxonomic assignment process, is that this was an unassigned sequence. So the ASV is believed to be real, but we couldn't get a hit in the, at least not a reliable hit in the reference database. And so I want to assure you that there is a mechanism for dealing with this so that the sequence can be shared, the data can be searched, and hopefully later updated as reference libraries improve. So here the scientific name is given as biota in Certesides, and then there's a specific worms identifier that's reserved for this case. And so this is a way that you can share your sequences even if you don't really have any taxonomy to apply to them. And then finally, getting more into the nitty-gritty of some of those molecular biology metadata. Again, I just want to point out that this is being linked through the occurrence ID. Here we have the representative sequence for the ASV, and I know I keep saying ASV. I want to assure you that this also works fine for OTUs. Zooming in a little bit, we have the DNA extraction concentration, the target gene that was sequenced in this case. It is possible, and in fact, Katherine did publish sequencing of multiple target genes from the same extraction. The target subfragment, the PCR sequences below this, the PCR the PCR primer sequences, the PCR primer names. Going down a little further, the reference for those PCR primers, the sequencing platform, and then here there's an area where there's a little bit of ambiguity, but I think it's something that I'm actively working to improve with these standards, and I would definitely enjoy talking with anybody else who has ideas about this, which is describing the clustering or the denoising algorithms, as well as the taxonomic assignment algorithms and reference databases. This is something that I think a lot of people would find useful if it could be broken out into better granules that could be filtered on, for example, confidence thresholds for taxonomic assignment. Okay, so you've seen a bit about how you can publish eDNA data using the Darwin Core standard. I didn't get into the EML standard in terms of sort of wrapping up that entire database, but it's very similar to ISO 19115 if you've ever used that to publish to NCEI, for example. And I think the take home here is that I hope I've convinced you it's a plausible and a useful system, but I want to emphasize that you don't have to use OBIS and GBIF. If you have other standards, if you have other platforms that you're using and they already interoperate or otherwise satisfy the international use of your data, then great, that's totally fine. But if you don't have those, don't start at the bottom of this mountain. Try to start halfway up by using these platforms. They're off the shelf. They use open standards. There's really minimal risk in terms of financial investment and staffing investment, and we're here to help you learn them. So if you have any questions, please reach out. I'm always happy to answer and collaborate on different projects. And I'd like to finish by saying thank you to Nicole for organizing this and to the Omics Working Group for the invitation. And then also thank you to Katherine Silliman, who I don't think I actually thanked for using her data. So with that, I'm happy to answer any questions.
Thanks, Stephen, for the great presentation today. Very informative and a lot to learn. Thank you very much. Let's go get into some questions. So our first question is from Paula, who asks, is there a difference between OBIS and GBIF compared to Pangea?
Oh gosh, I know Pangea, but I have not I have not worked with it enough to be able to speak confidently about it. So if there's a more specific aspect of it you could ask about, maybe I could give you a better answer.
Thanks, Stephen. We have one more question from Katrina, who asks a question about taxonomic identifications from eDNA in occurrence core. How can one include the original identification, i.e. from the reference library, and also potentially an omics taxonomy ID from NCBI, and or a traditional one like worms? So how can one include original identification?
Sure. So let me jump back to those slides at the end there. Okay. So there's a term here that I did not include, which is called taxon concept ID, and this is a place where you can use an identifier from example, BOLD or UNITE are two databases that are already working well with GBIF. I guess in that DNA guide that I shared before, it has specific advice for trying to include all of that information. And then I would also temper that by saying that this type of problem of working across taxonomic databases, depending on where the data are being published, is a clearly recognized challenge by the community, and people are working hard to try to accommodate all that information in one place. And so I guess the short answer is there are different terms that you can use depending on your exact situation. There may be some nuance in how you use those, but also too, if you have specific instances, feel free to reach out to me, and I'll be happy to work with you on it.
Stephen? I have a question, and that brings us, we have a few more, just one more coming in. But my question is, so what can we expect from future development of these two systems, and what exactly would you like to see, and do you plan to incorporate in future development of these systems?
Yeah, that's a great question. So let me go back here to emphasizing that right now we're in the middle of a 5-year period that is the second letter of agreement between GBIF and OBIS. And we recently, in February, had a meeting that I participated in where we spent a few days hammering out, actions that could be taken to remove any dissonance in the interoperability of the data, but also the user experiences of using these two platforms and working in two communities. For example, when you're doing analysis in the marine community world, frequently a starting point is to get all the data of your query from both OBIS and GBIF, and then sort of deduplicate and go on from there. So that's exactly the type of thing that we're interested in reducing is any dissonance in that experience. That action plan, like I said, will be coming out later this year. I can't promise when exactly, but I think it'll be something that a lot of people will be excited to see, that the two organizations have some formal plans to work to tackle certain problems. And then the next thing that I want to go to, jumping to this slide, things in development. I think there's a lot of low-hanging fruit where omics data can be made more findable and also more interoperable, and that's through things like sequence searches, which is obvious. You might not be interested in like I said, a taxon per se. You might be more interested in saying, where else has this sequence been seen? Similarly, the service of all the collateral data, all the ancillary data that goes with this type of experimentation, it can be a little bit difficult to coordinate all of it. So for example, I mentioned there's an extension for including your chemical or physical measurements of the environment. But the reality is that if you have a very large set of environmental data that is traditionally served up elsewhere, for example, through like ERDAP, it's usually best, or at least I think my default is that I would advise people to publish that through the way where it's traditionally published, and then use the metadata to link the two bits of data. And I think not just myself, but a lot of people would be interested in seeing that data served more seamlessly. And that's true of all biological data, not just DNA data. And then the other thing that I would like to point out is that there's a lot of discussion in the eDNA world about how do we make this data useful for people that are not necessarily technical experts or scientific experts in it. What are the limitations? How do we express it in ways that it won't be misused or misinterpreted? And there's really the community community of users, and the community of data generators is across a pretty large spectrum of both knowledge and technical expertise. So I think continuing to create materials to help people learn about these things, with multiple entry points where they can jump in and work with these organizations, sprinting if they have the technical expertise and scientific expertise already, or more gently if they're a beginner to the realm or only really interested in the data casually. And that's what this fourth bullet point can help do, is this eDNA publishing tool from GBIF. It's a place where a less technical user can feel comfortable working with their data and trying to share that data. And then the other end of that is with the data coming out, like I said, about the describing, the clustering, the denoising, the taxonomic assignment, and other choices that are made in the analysis. It's important that we be able to filter and query the data with those aspects in mind. And we're not there yet, but I think we're well on our way. It's clear, the importance of it is clearly understood, and so it's a matter of finding the resources to do that development, I think, for both organizations.
Mm-hmm. Undoubtedly. Thank you. Moving right along, we have quite a few questions coming in. So this one's from Jeffrey, who asks and says, ASVs that are not one-to-one, or multiple known species that have the same barcode, or multiple barcode sequences for the same species, will they be treated the same as visual occurrences from other methods? Or will GBIF/OBIS require certain parameters and reference databases of taxonomies submitted?
Ooh, that's a good question. I'm not sure, to be honest. I don't think I've considered that explicit circumstance yet, just because it's not something that I think is at the forefront of most data sets that I've encountered, most situations. I would say that right now the data are still being anchored to taxonomic assignment, perhaps more than would be preferable for the omics community. And so that type of choice would probably be made by the person publishing the data as to whether how they handle the taxonomy that's associated with these sequences. But I think, Jeffrey, that if you have, like, one of the ways that we make the biggest advancements is by tackling explicit use cases. The reason I use Katherine's data and that experience was because we learned so much working together to get that data published. Katherine's group has poked a lot of holes in what we do, and we've been doing our best to evolve to meet those needs. And so in the situation that you might be describing, if you have a data set where where we could explore that example, I'd love for you to share it with me, because I think it's a good question.
Thanks, Stephen. Our next question is from Kelly, who asks, what is the connection or communication between OBIS and GBIF, US, and NCBI?
That's a good question. Right now, it's rather minimal. We're a small staff, and I think, like all small staff, to some degree, your network is driven by circumstance and your pre-existing networks. I will say, though, that I personally do attend meetings for the Genome Standard Consortium to try to improve the mix-S standards, and NCBI is also involved there. And so I'm making a concerted effort to try to develop our relationships. I'm well aware of the fact, and I think NCBI is well aware of this fact too, that the gap between the experience of publishing to NCBI and publishing to OBIS, for example, there's not enough streamlining, there's not enough sort of conscious synthesis of those two workflows. And I think that's something that we could improve, and I'd very much like to continue to do so.
Thank you. Our next question is there room to include confidence levels for DNA taxonomic names, either the scientific name or all of them?
Yeah, so let me see if I think we have sort of an example of this in Katherine's data here. Let's see, so there it's in identification remarks, but also, is it here? No, I think it's just in this identification remarks. So here, there's this confidence expressed in these identification remarks, but I don't think that's a good, or rather, I think we can come up with a better solution. And that's specifically what I was talking about, and is actually something where I think on my to-do list is pulling an issue to begin talking about this in the within the Genome Standard Consortium world. People recognize that we sort of reached this next level where it's like, okay, now people really do want to reuse data and be thinking about, how valid are those taxonomic assignments based on things like confidence and methodology. So I could see this type of information being able to be broken out in explicit terms in the near future, but so far it's generally included in a place like identification remarks or down here where you're talking about the denoising or other assignment other assignment related terms.
Awesome, thank you. Artie, we have one last question today. It's from Olympia, who asks, thank you for the presentation. I wanted to ask if there are pipelines that are recommended to follow for that type of data due to high complexity of eDNA. And before I hand it to Steve, the answer to this question to Steve, I would consider the NOAA Omics Data Management Guide. It's a great guide that was recently published by NOAA Omics and research staff. And it has a great intuitive outline and guide for addressing the management needs of eDNA data. But with that, I'm going to pass it back to you, Stephen.
Awesome. Yeah, Nicole, I have a big smile on my face because our heads were in the same place. So you can see here under guidance, I am listing that NOAA Omics Data Management Guide as a great place to start. Like I said, we try to work with as many different groups as possible. And so, for example, OBIS USA staff, we do meet regularly with people from AOML and PMEL, KBase, the Smithsonian. In USGS, we have an eDNA community of practice. In the MBON world, we're talking about this. And then, of course, internationally, there's the Genome Standard Consortium, the various European projects that are tackling a lot of the same challenging questions. And so there are a lot of guides out there. But I think the NOAA Omics Data Management Guide is one great place to start. I also, again, I would urge you to check out this GBIF published guide, which has an OBIS specific section in it. It gives, I think where this guide will be most useful is if you're not very familiar with biodiversity data and working through these platforms, it'll help you make the connections between your DNA data and how that corresponds to other biodiversity data. And then finally, I also want to say that the IU's Biodata Guide, which is something that's developed in part through this standardizing marine biological data working group, or SMBD. This is really a very sort of boots on the ground, like community driven biodata guide, where let me go to a couple of these data sets. So for example, this data set here from Mbari, was developed and documented through there, and that community, this SMBD working group, has been really useful to a number of people who have published eDNA data and omics data too. So for example, I know that this CO1 data that was published recently the person who published it did attend a workshop that we ran as the SMBD, and that was something that I think they found these guides useful. Beyond that, there's also, I had mentioned there's a place where you can reference the Darwin Core terms that's down here in the bottom. So you can sort of browse those and explore them. I think that user experience there is a nice one. It's very, very straightforward. OBIS also has a manual with a lot of information where you can get more into the nitty-gritty and answer difficult questions. And then GBIF also has a manual that is not just specific to publishing, but also working with the IPT. Let's see, beyond that, there's, in this training category, there are some videos that would probably be very helpful to a person who is starting this from the start, or starting this with relatively little knowledge. And then I guess the other thing I would say is, make an effort to start with a data set that's simple. Don't come at this with like your most complex data initially. Try to find something that's a little more bite-sized so you can get a feel for it, because the initial learning curve can feel a little bit steep, but once you get over the basics and get a sense for how Darwin Core works and how these publishing platforms work, then I think it's fairly easy to run and to start to really chew on those difficult questions about what is the best way to describe and share your data.
Awesome. Well, thank you, Stephen, so much for your very informative seminar today. Thank you all who joined online, and we will see you next time. Our next seminar will be May 15th at 2 p.m. Eastern Standard Time on gene expression responses of stony corals to ocean acidification from shallow to mesophotic reefs. So very excited for that one in May. Thank you all very much, and thank you, Steve, for your great presentation today.
Thank you.